So Rami, Aqeilan, we're very proud to have you here in our Neurodevelopmental Hub. Thank you for coming such a short notice and really starting our first Hub lecture for this year. And now in terms of your CV, a true international scientist, started BSc in Jordan and then moved to the Hebrew University for master's degree in G, and then to Thomas Jefferson University in the US for postdoc. Then you started your career in Ohio State University, and luckily for us, came back to Israel. Most of your career, if I understand correctly, was centered around cancer biology and the WAX gene. You numerous awards, recently the ERC Coordinator Grant, and author more than 100 papers, 117 if my PubMed search was correct, and additional awards in cancer biology. And I'm really excited to hear your talk today and the connection between cancer and neurodevelopmental. I think it would be a great perspective. Thank you.
Thank you, Moran. Really an honor. And thank you, Karen, for the invitation. I wish we could do this face-to-face, but it's a start. So, okay, so today I will be talking to you about our new research venue that we started in the lab a few years ago. And this is really to study neurodevelopmental disorders, in particular those that are associated with the WOX gene. This whole study get us to know more and more about rare genetic diseases, and I thought to make a very general introduction about this whole area by having some questions and answers, just to make everybody on the same page. I find it very interesting about these questions and answers. From there, then we can go into the outline of the talk and dive in more into our research work. So, as I said, I will do a few true or false questions just to make everybody on the same page. There are over 7,000 rare diseases, with more being discovered every day. You can answer to yourself if this is true or false. The answer is true. In the US, a condition is considered rare if it affects fewer than how many persons combined in a particular disease group. You can choose A, B, C, or D, and the true answer is B. It is 1 to 150,000 births that consider a rare disease. There are also ultra-rare diseases that even go below these numbers, but this is how it can get rare. Rare disease affect how many million people worldwide? A, B, or C, or D. And the true answer is actually 350 million people in the world. They live with these rare diseases. If all the people with rare diseases lived in one country, it would be the world's 6th most populous country. What do you think? I think it is false. It is the 3rd most populous. Only 30% of rare diseases have an FDA-approved drug treatment. That's wrong, because only 5% have an approved drug treatment, and approximately 50% of the people affected by rare diseases are children. True. Rare diseases are responsible for 35% of death in the first year of their life. True. On average, it takes most rare disease patients 5 years to receive an accurate diagnosis. This is really a pity, but the answer is false. It takes 8 years. This is improving with time, but this is roughly worldwide. 30% of children with rare diseases will not live to see their 10th birthday. Unfortunately, this is false because it is the 5th birthday. most of the children will die within this time period. And now, just to sum this up, we know that all these facts, and what really stunned me is the fact that that if we talk about 83% of the rare genetic diseases affecting children, about 90% of these pediatric rare disorders comprises the nervous system. So the CNS is mostly affected by these rare genetic diseases. And in particular, we are interested in our lab to study developmental epileptic encephalopathy, which is responsible for almost 40% of the seizures that happen in children within the third year of their age. And you can see here this figure showing the different genes with the different effects they do. You can see here the WOX gene that we will talk about it, and it is called more developmental and growth defects leading to DEE, and those other genes contributing to these particular type of epilepsy. This is a summary that we will talk about today. So I called it our work in Aqeilan, and this is an outline that I will be covering in the next few minutes. We will talk about our interest in common fragile sites. We'll talk about worry and SCAR12 syndromes, how we modeled them in mice, the gene therapy that we started, how we used brain organoids to better study these syndromes, and we will conclude with some maybe an overall conclusions and take-home messages. But we were interested in these developmental epileptic encephalopathy, specifically worry and SCAR12, because of this involvement of a gene that we worked a lot in the lab that is called WOX. But how we did arrive to this gene, and what is WOX, and what is these sites that we are interested in. Before starting, I just want to introduce you to the team, the neuro team in the lab. Sri is a postdoc, Daniel is an MD PhD student, Sarah is a master, and Bara is a master student. Okay, so common fragile sites, and what are they, and why we are interested in studying them. So these are loci, specific chromosomal loci that are prone to form visible gaps or breaks on metaphase under conditions that leads to replication stress. Replication stress, you can imagine this as cells that are undergoing very fast DNA synthesis. This happens also in cancer, but also during embryogenesis. And due to falling of the replication fork, there will be these gaps and breaks that we see in the chromosomes. They happen in a very particular regions in the genome that are present in the genome. They can be induced in vitro by several types of drugs like afidicolin, which is an inhibitor of DNA polymerase, but also others. They can be induced by overexpression of oncogenes. And more studies are showing that they can also be responsible for de novo mutations. Now, the first fragile gene, these sites, they are encompassed by genes, and these genes are large genes, very large genes, sometimes spanning more than 3 megabases in the genome. The first gene to be cloned, the first fragile genes, was the FIT genes, cloned in Kay Hubner and Carlo Croce's lab, where I did my postdoc in Thomas Jefferson University. And later on, we started working on this second fragile genes that was discovered, the WOX genes, standing for the WW domain containing oxidoreductase. And as you can see, much of the work was really associating these genes with metaplasia, dysplasia, loss of heterozygosity, translocations, homozygous deletions, basically in cancer, because they showed this loss of function in cancer and several types of cancer. But we want to talk today about the brain, and I will focus our topic today about these common fragile sites and gene products of common fragile sites in the brain. So why did we really study and we become more and more interested in the brain? This is a study that was published 2 years ago showing mapping of these common fragile sites using next generation sequencing. And it shows mapping of these several genes, as you can see here on the right, on your left, showing many of these genes that what is common about between them is that they are associated or involved in neuropathologies or in pathways that are important for the CNS homeostasis. You can define here some of these protocoadherents, PCDH17, PCDH19s, the CTNND2, and CDH12, and so on and so forth. It seems like many of these genes are large genes, but they are also candidate common fragile genes that are involved in those pathologies, neuropathologies that we know of. As I said, today we will be focused on the WW domain containing oxidoreductase, the WOX gene. As I said, it's a gene product of a common fragile site in the Fra16 region of the long arm of chromosome 16. So how did we really start getting interested in all this area of neurodevelopmental disorders? It was because of this study that was published in 2014 on a Israeli-Palestinian family in the north that showed that there is a loss of WOX function in children with this disease that is now known as SCAR12 or spinocerebellar ataxia type 12. And this is because of a missense mutation leading to a truncation of WOX at very early on at this arginine arginine number 54. Another family was also published in this paper where the mutation is in the C terminus of WOX at G372 converting to R. Again, a missense mutation leading to very reduced amount of WOX expression in these in the cells. It's a germ line, and these children were showing to have epilepsy, ataxia, intellectual disability, all symptoms and phenotypes that are associating this mutation with epileptic encephalopathy. So that was the first clue that germline mutation of WOX could lead to a neurodevelopmental disorder. After this paper, there was an avalanche of studies that linked WOX loss of function, these variants and mutations in the WOX gene, with what we know today as the developmental epileptic encephalopathy 28, or WOX-related epileptic encephalopathy, in short, worry syndrome. This form of the syndrome is much severe, resulting in lethality of most of the children with the mutation very early on in life. The seizures and the epilepsy appears in these children really very early, like the same day of delivery or next day. One report also reported in utero seizures and death of the infant or of the embryo. So what do we know about this genotype-phenotype correlation in these syndromes? We know that worry is mostly associated with copy number variations leading to loss, deletions of the genes, sometimes complete deletion or partial deletion. And SCAR12 is more associated with those point mutation, missense mutations that are leading to much milder phenotype in relative. Still, these CAR12 patients, they are really in very bad shape. Nonsense mutations were also associated with worry. Basically, the more deleterious the mutation is, we get a more severe, and the clinical phenotype would be more linked to worry syndrome, whereas a milder phenotype would be associated with SCAR12. With time, now this is a review that we wrote with Ehud Ban, who's currently in Wolfson Hospital, and it shows that more neurological disorders are also linked to WOX mutations. We know of cases with multiple sclerosis, autism, Alzheimer. Recently, Parkinson's disease was also added to the list. So it means that this gene, this WOX gene, is very central in regulating CNS homeostasis, and its defect, loss of function, either in a heterozygous form or a homozygous form, it will lead to some kind of neurological disorder. Today, our focus will be on epilepsy, and I don't have to tell this forum that epilepsy is this very common disease. Almost 70 million people in the world have epilepsy, and it happens due to this imbalance between excitatory and inhibitory neurons. It can be as a result of genetic modifications, as we will see the WOX gene, the GRIN, other genes that are involved, but also it could be caused by trauma or brain injury, and so on and so forth. Now, WOX is already in the panel of epilepsy gene discovery. So whole exome sequencing or targeted sequencing for WOX gene is there. So we are now aware of hundreds of patients in the world. It can go under this ultra-rare disease or syndrome, but more and more are discovered. Unfortunately, as I said, most of the patients with this with worry syndrome would die really in between 1 and 4 years old. So what do we know? Once we have this all knowledge, and this was an emerging, in fact, some families also approach our lab. We worked on the gene, as Moran said, a lot on the cancer biology context, and we have generated many reagents. Basically, our lab is generating a lot of mouse models, but also tissue culture models. We analyze a lot of samples, clinical samples associating or linked to cancer. So we wanted really to see if we can contribute anything to this emerging phenotype associating with WOX loss, and see if we can help in this manner. So this is the mouse I generated when I was a postdoc in Carlos' lab back in 2007. This paper was published. We knocked out the WOX gene, hoping that we can see more tumors. The mice were very poor. They have growth retardation. They had seizures, but I really did not thought that this is neurological. I thought they had a hypoglycemia, and I thought that this is the reason. They also had ataxia. They looked like more aging, and they die within 4 weeks. So 100% of the mice would die with a very severe metabolic disorder. So when we wanted to look more carefully at WOX function in the brain, we went back and started looking at where is WOX expressing and at what time during development and during the adulthood, and where WOX expression is rising. And as you can see here from the HPT website, you can see that WOX expression is very high during very early embryogenesis, then it goes down, but then it peaks during adulthood. And it is expressed widely almost everywhere in the brain. This is a figure that was done by Daniel, and it can show that WOX expression is everywhere in the brain, everywhere in the different brain regions, the cortex, cerebellum, in the hippocampus, also in the murine brain, even in the organoids, as we will see. So three in the lab decided to figure out where is the critical function of WOX in the brain. And this is why he used this mouse genetics in order to breed our WOX flox mice with different Cree models that drive the deletion of WOX somatically indeed, but in different compartments. So he used Nestin Cree for the neural stem cells, Synapsin 1 Cree for mature neurons deletion, Olig2 Cree for oligodendrocytes, and GFAP Cree for astrocytes. And as you can see here and appreciate, when deleting WOX in the neurons, in the mature neurons, or in the neural stem cells, it will phenocopy what we see in few slides before when we have the null mice, where we have the germline mutation in all the cells in the body. Deleting WOX in oligodendrocytes or in the astrocyte did not really affect this phenotype of epilepsy and seizures and growth retardation that we saw with Synapsin 1 Cree. This paper was published recently, and we concluded in this paper that deletion of WOX in neural stem cells or in the neurons is really responsible for this hyperexcitability that we see in the mice. We also define many of those molecular and cellular changes. In particular, I will just mention this hypomyelination that we saw, and we kind of characterized briefly in the paper. We're now working on this much more in depth, showing that there is this non-cell autonomous function of WOX in the neurons affecting maturation of oligodendrocyte precursors and leading to their defective maturation, resulting in hypomyelination. This is also connecting to our work on multiple sclerosis, which I will not talk about today, but now we are following, Sarah in the lab is following this work and more learning about WOX function and oligodendrocytes. Okay, so with this knowledge that we knew of now, that neuronal WOX is really responsible for the whole phenotype that we are seeing in the mice. Sri asked in the lab whether we can restore somehow the phenotype of wild type by restoring the WOX gene expression in the neurons. So how can we do this? So we did this using adeno-associated virus expression, using a very particular and specific promoter, the synapsin promoter, to drive expression now of the WOX gene in the mature neurons. Sri injected these in P0 neonatal mice in this very small window in P1 or P0 in the ventricular zone, and then waited to monitor what happened to the mice. The results were remarkable. I did not believe this at the beginning. I asked Sri to genotype those mice that he injected several times in order to make sure that they are really knockouts. But with rescue of WOX in the brain, and we injected this ICV, as I showed, in the brain, single injection, and this led to gain of weight. The mice survive. We have some mice now that extended to even 1 year and beyond. The paper is published, and I will not get into the more details into this. Just to show you and share with you, this is the electrophysiology study that we did with Dr. Shanice Term from Haifa, and showing the hyperexcitability of neurons compared to the wild type, and this is how the rescue would look like. This was done by cell-attached recording. So this proof of concept for gene therapy, we were very happy to see that it is achieved by this AAV9 Synapsing WOX single injection. At present, this is now being developed by Massey Therapeutics in order to proceed to clinical trials in patients. And we were very lucky that we won also the cover page of on Biomolecular Medicine 2 months ago, where they showed this decoration of myelin in the wild type and the reduced in the knockout and the restoration with the synapsin expression of WOX and leading to more and more myelination. So this is just to summarize this first part. So I showed you that WOX germline mutation causes SCAR12 and Wari syndrome. Neuronal WOX deletion in mice recapitulates WOX germline mutation. WOX loss results in hyperexcitability, astrogliosis, and hypomyelination. Much of these details are in the papers. And that neuronal restoration of WOX rescues WOX null phenotype using gene therapy. Okay, so now we wanted in the meanwhile also to study this defect of the CNS, specifically of the epilepsy that we see in these children using a human system. And this was coinciding with this emerging methodology that was developing in the recent few years by establishing the organoids based on the use of stem cells or induced pluripotent stem cells that can organize into a three-dimensional culture, and that could be very similar, sometimes identical to human tissue of term of histology, morphology, developmental stages, and even functionally, as we will see. In particular, we were interested in these cerebral organoids. This is just to remind that this organoid was chosen to be the method of the year by Nature Methods in 2017. So a lot of attention was given to this methodology. And one of the facts that really drive us to go in this direction is that we could really faithfully start to look at what happens during embryonic development. So we are talking about neurodevelopmental disorder, and it has been shown that there is this very close similarity between, specifically in the second trimester during the embryonic development, between the organoids, the brain organoids, and what happens during embryonic development. Of course, this methodology, you would see many papers nowadays summarizing the success of the organoids in modeling the homeostasis, but also diseases that are associated with the central nervous system, and those different aspects that it can be of advantage to use the organoids relative to all these other animal models or tissue culture models. But as every method, there is also some disadvantages. So I will mention here a few of the pros and advantages to use the brain organoids. And really, this is allowing this study of complex and relatively inaccessible human brain tissue. When studying the mouse brain, it's really very different from the human brain, specifically when you talk about the complexity, the number of the neurons, and the synapses that is going on in the human brain. It can give you an intrinsic capability to generate cortical-like and cerebral-like tissues, and this is one of the biggest strengths of this method. It mimics much of the developmental stages, as I show in the previous slide. That'll give us really the advantage of modeling neurodevelopmental diseases. It can be generated in a pattern manner where we can look at specific brain regions, let's say the cortex versus the cerebellum, or unpatterned organoids where we can look now at the different regions of the brain. Even nowadays, there is many data or many papers showing co-culture of these brain organoids with other tissues that are coming from different lineages, like co-culturing of brain organoids with microglia or even endothelial cells to generate blood vessels. There were attempts to use these or to transplant these organoids in the mouse brain and show how would they develop further when they get vascularized in the brain of the mouse. And of course, using them as a platform for drug screen, as you will see for gene therapy in our case, to allow improvement of precision medicine for developmental epileptic encephalopathies. However, there's also some limitations. We're still talking about a culture. These organoids would grow to some limit where now there's limitation of the nutrients diffusion, resulting in necrosis in the core. So some studies now are showing that people are slicing these organoids at certain time, allowing the nutrients to diffuse in more and overcoming this limitation. So there is a lot of development in these techniques and methodologies nowadays, but still there are issues with how much these organoids are heterogeneous and reproducible, because they are still under culture conditions. Sometimes you see hypoxic stress, chaperone proteins that are induced. And a main thing which is for our work is not really a kind of a disadvantage, but it reflects those early stages of embryonic development, though there are now more and more long-term culture showing that these organoids can also reflect postnatal transcriptional signatures and functions of the brain. As I said, there's an issue with reproducibility. Again, this is a culture, and this is why we do a lot of validation, not only in mice, but also with the organoids themselves, with the neural stem cells, and so on and so forth. But no doubt that they are a great tool to analyze or to start looking at things that happen really in the patient and not cannot be modeled in the mouse, for example. So what are we talking about? So how do we do this whole thing of brain organoids? Usually we start with an adult pluripotent stem cells or even an embryonic stem cells. The cells are grown, then they are allowed to form an embryoid bodies. Within a week, then we switch the media to start a neural induction. And we start to see these neural ectoderm lineage specification, and then the media is changing to differentiation media in order to allow for the expansion of the neural epithelium. And here the these tiny organoids would be put in a matrigel in order to give them more coherence and to allow kind of mimicking of a basement membrane where they can grow and mature in a better way. And then these organoids will be put in a shaker on a bioreactor in order to help allowing the diffusion of the nutrients and the growth of these cells and the differentiation of the different cells. As I said, here we are using unpatterned. So this is the cerebral organoid, allowing the formation of different regions of the brain. You can see here, for example, pigmentation that mimic or resemble what happens in retina development, but we can see cortex, we can see markers of hippocampus, and so on and so forth. But again, there are protocols that they are modeling patterned organoids where we can look specifically at very specific regions in the brain. So the ones that we use in our lab is basically either from reprogramming that we do from the patients or by genome editing using CRISPR, is either cerebral organoids, which is again unpatterned, giving rise to those different regions, or we also use oligocortical spheroids in order to enrich for the oligodendrocytes and study the myelination defects that we see in the patients. So for the reprogramming, we use the very standard Sendai kit reprogramming. And again, this was done on wild type, but also on WARI or SCAR12 patients. And once we have the iPSCs, they are validated by karyotyping, but also by staining for staining for pluripotence markers and for generation and the ability to generate teratomas. Then we start the protocol of the brain organoids. Here you see cerebral organoids of wild type human embryonic stem cells, WOX CRISPR and knockout, and restoration of WOX in the AAVS locus. So this is, I think, at week 10. And you can see a marker for SOX2, which degrade the progenitor cells, a marker for neurons, TUJ1, and the WOX staining, showing WOX expression really decorating these ventricular zone and what we call and know as the radial glia, as marked here by this CRIA staining, suggesting that WOX is expressed in these very early progenitor cells. The knockout doesn't express. If we now restore WOX expression in those organoids, you can see that it mainly expressed in these ventricular zone regions in the radial glia and the progenitor location. So we can already start to learn that, as I showed you earlier, WOX expression is very high during embryogenesis, during brain embryogenesis, specifically in the neural stem cells, and this could be validated now in the cerebral organoid protocol, even upon overexpression. Another thing that we did with using these organoids is really to look at their hyperexcitability. And as you can see here, again, this work was done with Shani Stern in Haifa on the different patients. This is the father, this is the mother, where they express very low levels of WOX. Again, in this particular localization, this is the son who's sick, no WOX expression, and this is when we overexpress using the AAVS locus. There is, by cell attached recording, the sample tracing showing more firing in the son compared to the parents. And when we restore WOX with this expression and the AAVS locus, we can rescue this firing back to the normal state. So even by electrophysiology, these organoids could tell us that we can assess the epileptiform bursts in the patient, and we can monitor whether maybe expression or re-expression, genetic expression of the gene would rescue, or even with the screen of drugs that we are doing in collaboration with Peter Carlin. Another phenotype that we also see using these organoids, which also coincide with what we saw in the mice, is this astrogliosis. Now, the beauty of using these organoids and using this from stem cells or from a pluripotent stem cell is that these cells, when they are allowed to go to the ectoderm lineage, they would differentiate into the different types of cells. It is not only one cell types. So we get the neurons and we see the evidence by TUJ1 expression, but also we start to see astrocytes. And you can see here, this is staining for GFAP and the S100 beta markers of astrocytes. You can see that the knockout expressed much more of those markers in 15 weeks and in 24 weeks. This is by immunofluorescence, and you can see here by qPCR at the different stages. And when restoring WOX, again, we would reduce this amount of staining and amount of these astrocytes that are generated. Now, these organoids could be also very helpful to study molecular changes. So we did RNA sequencing. And again, the details are in the paper. I will just give you here some directions that we are following. We saw using gene set enrichment analysis that there is a change in the regionalization. So regionalization means that the regions, the pattern of the cortex could be different in these knockout or patient-derived organoids. Indeed, we saw that one of those major pathways that control regionalization is the Wnt beta-catenin pathway. And we saw that in the knockouts, we got really very high expression compared to the wild type in several of these Wnt genes and target genes that are regulated by Wnt. Here you can see this qPCR on a different batch that Daniel generated, and also looking at beta-catenin accumulation in the nucleus compared to the wild type. And using this follow-up of the different weeks, we can see that there is a chronic activation of Wnt genes, but also the target genes with time that we followed these organoids. Bara in the lab wanted to assure that this is really happening in cells. So she used this neuroblastoma cells, the SY5Y. She knocked out the WOX, and she again could see that there is increased levels of Wnt genes and Wnt target genes upon WOX deletion. So WOX regulates or negatively regulates the Wnt pathway. This was also validated in our cancer models that we studied previously. So we are kind of sure that this regulation of WOX and the Wnt, or this functional crosstalk, could really contribute to the phenotypes that we saw. So Daniel went back to the organoids and he started looking at these different regions in the developmental cortex. And this can be monitored by staining for these different markers that marks the different layers of the cortex. And he could show also by looking at real-time PCR or by looking at immunofluorescence that the knockout really suffer from reduced maturation of, for example, layer, the upper layer that is stained with the SATB2 compared to the wild type, and restoring WOX again would allow this regionalization to be somehow fixed. And this is the quantification of these figures. So yes, we can use these organoids also to study the molecular aspects of WOX regulation or any gene that is involved in these neurodevelopmental disorders. So now, following these results, Daniel wanted also to look at what would happen if we use now the gene therapy, the AAV9 virus that we used in the mouse, on these organoids. And this he used now in order to see if we can rescue the phenotypes that we see in the organoids derived from the patients. And again, when we looked at the regionalization defect, this is the wild type. You can see that the knockout has the defect in SETB2 and CTIP. You restored with AAV9-WOX, you can restore this regionalization defect. Doesn't happen in the SCAR12, which we also noticed previously. However, it is fixing the defect that we see in the worry, the more severe phenotype. So it seems that this regionalization defect that we see, and we think it is somehow regulated by the Wnt pathway, is relevant to worry patients and not to SCAR12 patients. So to summarize, I think that this data told us that whether we are using stem cells or embryonic stem cells that are edited for WOX or that are taken from patients, we can generate these organoids that can help us to understand the physiology of what is happening during embryonic development of the brain. Also about those changes that happen not only in the neurons, but also in the cells that support the neurons. For example, the defect that we saw in the astrocytes. We looked very carefully on DNA breaks because WOX is a direct activator or directly involved in DNA damage response. And we saw that there are more breaks. And we have some hypotheses, how does these breaks really contribute to the phenotypes? We're still testing this. We also see some molecular pathways that we are changing, Wnt pathway, for example, and how it leads or how it might lead to these defects in regionalization, for example, this cortical dysplasia that we found in the worry organoids. Now, as I told you before, the mice showed hypomyelination. The patients showed thin corpus callosum. So Daniel and Sri wanted to model this in the organoids. So they used this oligospheroids that give advantage to the oligodendrocytes to develop early, a very long protocol. It takes about 30 to 38 weeks. But they could again show that in the knockout spheroids, this is how they like to call them in the literature, we see much less myelination as stained by myelin basic protein. And even Sri did an electron microscopy to show again that although the wild type has myelination sheath around the exons, there is much less in the knockout. Again, telling us that these organoids could really faithfully resemble or phenocopy what is happening in the brain of the patients and consistent with what we see in the mouse models. So this is just to sum up everything. I think we spent a good amount of time to model WOX using mouse models and cell culture, including the brain organoids. And we kind of seeing that these combination of these different models could lead us to better understanding of the cellular and the molecular mechanisms and the physiological mechanisms behind this disease. We think this is an excellent platform in order to further later testing for therapeutic intervention, whether this is gene therapy or RNA therapeutics or even drug screens. Hoping that in the future we could really, and this is what we are actively doing now with MASI, doing testing in NHP and planning our clinical trial to try to help the children affected with WOX mutation. Maybe we have just a few more minutes to sum up. I think I talked about this intensively where we discussed after each of the results and findings I showed and shared with you. This is just to again share with you and acknowledge the whole group working on in my lab, particularly those 4 people that work on the neural and the CNS, and acknowledge the funding and our collaborators, Peter Carlin and his team in Toronto, Ori Pelis, who helped us with the oligodendrocyte maturation studies at Weizmann, Shani in Haifa, Udeid here at the Hebrew University, Yaqub Khana taught us how to do reprogramming, and Vicky, I less mentioned her work, but associating with the triple negative breast cancer. And thank you